What's up, Mindsetters? Welcome to it. It is a Wednesday, and you're hanging out with Looney and Cheryl.
Welcome.
How are you?
I'm well, and you, Lily?
I'm good, thanks. So what are we doing for the Mindsetters today?
Oh, we're doing wonderful food tests. We're going to do a little bit of practicals. So we're going to
Awesome.
mix some chemicals up.
All right, Mindsetters is going to be very exciting. I can see some stuff there that you can't see, but you're going to see now. Just so you know how to hit us up, Facebook and Twitter at Learn Extra. And then you can download your show notes, your videos, and your schedules on learn.mindset.co.za. Just a reminder to all of you guys, we do have something running on our Facebook page. All you need to do is to show the love. I like to do this. Show your love. So what it is that we're celebrating the month of love. But like I was saying to the Mindsetters yesterday, you don't need to show love in Feb only. You must show the love the whole year. So all you need to do is upload a picture of yourself and that special someone or a special thing. For example, your dog, your cat, your hamster, anything with the hashtag show your love. And remember, guys, you must be in the picture, and then right at the comments block you need to tell us why it means so much to you, and then we'll choose it, and it might be on TV, and then you'll be famous. So do that, show your love, guys, by posting those pics on Facebook. And now we're going to get straight into the lesson. So Cheryl.
Are you listening to that, hubby? Show the love.
Yes.
Welcome, grade 10s. Those of you who followed us last week, all right, we started off, we're looking at biochemistry, the chemistry of life, and we looked at all the different inorganic. We looked at water, and we looked at nutrients, and we looked at carbohydrates, we looked at proteins, we looked at fats, and we looked at enzymes. And today we're going to look at the practical ways in which we can test, right, if those are positive, if those are found, and we're going to call them in different food groups, right? We're going to do a few practicals with you, and hopefully maybe your teacher does it as well, and it's a fun portion, all right, where you just can have a little bit of fun and mix it a little bit up, okay? But very, very important, right? When we say, yes, it's fun, this section of work is exceptionally important. For those of you taking life sciences here, what the CAPS document, I'm sure you've heard about it, they want to put in practicals at the end of the year as part of your exams. Those schools who do IEB exams, they do have practicals at the end, right, where your practical actually forms part of your paper. It's a fun part. I won't say I won't have to study, but it's the practical part of it. So you guys need to be able to, it's a skill, being able to do a practical, all right, in life science is actually part of what life science is actually all about. It's not just always cutting up hearts and whatnot, those kind of things. Okay, so the principles today are exceptionally important. When we do the food tests, they form the basis of so many of the practicals that we can do next year, right, when we do excretion, etc. So it's just not, right, we can't limit it just for today. So if we have a look, right, all that we're going to do is I'm going to test, right, for different food stamps, and then those tests, you need to be able to use it. It's not the case that I can test for glucose. It's a case of I can test for glucose. I can then, because I can do that, I can test for anything, all right, and see if glucose is present. So we're going to move across. So you're going to just chat.
Chat. I'm good at that. Mindsetters, just to clarify stuff, I see on the Facebook page that most of you guys have been just posting on our wall, and then you don't show us a picture. What we want you to do, for example, I make an example now, okay? If I post a picture of my nephew and my niece, like a selfie with my nephew and my niece, that means I'm showing the love, and then I'll say, I love them because they teach me so many things about life, and they show me a different perspective about life because they're so tiny, and I see the world through their eyes, like, you know, sweet stuff like that. So that's how I'll hashtag my picture, show the love, then I'll comment. And then just another thing, quick shout out to Samge and Mishak who are already tuned into the show. And make sure, guys, if you have any friends who are in matric who want to write the SAP exams, we do have SAP school, guys. It runs from Monday right up until the end of the week, and it starts at 9 o'clock. It takes a break when we have the live shows, carries on from 7 until the night time. So make sure, guys, you tell all your friends if they want to SAP their exams from matric last year. We have got their back, and we have support all the way. Make sure you don't forget to hit us up on Facebook and on Twitter at Learn Extra, and you can get your notes, your videos, and your schedules on learn.mindset.co.za. And if you ask Looney, but if I missed the show, what can I do? Don't you worry, we do have a YouTube channel. All you need to do is go to YouTube, and then you search for Mindset Learn, and there you will see that we have all our shows there. And if you have DSTV, oh, obviously you have DSTV, check your schedule for the weekend. All these shows are repeated during the weekend as well. Are you ready, Cheryl?
I'm ready. I just realized now I forgot to give the challenge question. So
Oh,
you know what
okay.
we're going to do today? We're going to just shuffle things around. So I'm going
Right.
to do the practical and then we'll do the challenge question. just
Okay,
now.
cool.
All right. Welcome, grade 10s. As you can see, I have a few things in front of me here. What we're going to do is we're going to test for, all right, I'm going to start off with, we know that whatever we're testing for is found. So I'm going to start off with to test for glucose, all right? So I'm going to use glucose. You're going to see how we can apply it later, but there we go. You can see we're going to put glucose in, all right? And those of you from last week, you would have remembered that glucose was sweet to the taste, all right, and it was soluble in water, if you can remember your monosaccharides from last week. So I'm just going to take some water and mix it here, and then we're going to make a nice solution. Okay, so now we know this has got glucose in, but that's not what we actually want to do. What we're looking for, all right, is how do we know that it's got glucose in? What is the test? Now, for each test, you need to know the following. One, what am I using? My aim, first of all, there to test for glucose. Then comes the second thing. For each of them, we are going to have what is called a reagent. All right. Now, when we come to this, the glucose, you might have a different thing at some schools. I'm going to use Benedict solution, which is blue. Some of your schools, you might have the combination of fillings A and B. Right, I know I should be measuring this out all nice and neatly, but we're just doing it for the show. Okay, now what do I talk about the chemical reagent here? All right, what I have now is I have glucose and I've added Benedict solution. All right, now what I want to do is I want to speed up the reaction. What do I do is I take it, and you can see here it's like a cooking show today, all pre-planned. We take our glucose in our test tube and we put it in a water bath. And what a water bath is, you take boiling water, right, you put it in a beaker. If you want to keep it boiling, you can put it underneath a Bunsen burner. And after a while, all right, now I want you to have a look. This is what happens. Okay, now this is the rule. I started off with blue and it changed color. If my reagent changes color, whatever I'm testing for is positive. Okay, so I used glucose. I know I used glucose, all right? I said to you, I know it's glucose. I added Benedict solution, that is my reagent, all right? And then I put it in a water bath, I heated it up, and my result, all right? Now, listen carefully, your teacher, you might need to use the word orange, or you might come across the word brick red. Okay? So when it comes to the experiments, these are the things that you need to know. Number one, what am I testing for? Glucose. Number two, what is my reagent that I'm going to use? Here we use Benedict solution, which is blue. At the end, what is my result? My result is a brick red or an orange solution. Right? If it's this solution, I can then conclude that there is glucose. That glucose, I have tested positive for glucose. All right? So now how do I apply it? Now, instead of using glucose, I can use anything. So I might want to take some grape juice. I might want to take some orange juice. I might even want to take some milk. I can now take any food, all right, put it in a test tube, add some Benedicts, put it in, all right, the water bath, and if the color changes, right, you'll see it goes from a blue to a light green, and then it will eventually come to this orange. If it changes to orange, whatever you put in that test tube, whatever food you put in, all right, it has glucose in it. All right, so there we go for the glucose one. Remember, that's part of the carbohydrate group. In front of me here, we're still testing for carbohydrates. All right, I have put, let me go over here, I know what it is. It's starch. All right, it's a starch, it's a polysaccharide. And what am I going to use? So the aim is to test for the presence of starch. For glucose, what was my reagent? Benedict's. What am I using when it comes to starch? I am using iodine solution. Now, if you can see, if I bring, all right, some iodine up, iodine is brown. If I put it on the side over there, I don't know if you can see it, all right? Iodine solution is brown, but if I add it to my starch, oh, okay, here we go. If I add it to my starch, I'm going to be, oh, there we go. I'm going to show you the result now. What do we have? All right, it goes blue, black. All right, it goes blue, black. Sorry, me and my earpiece are fighting today. All right, there we go. So we have a blue-black solution. So it was brown, and when I put it with starch, it goes blue-black. All right, so now that shows me that whenever starch is present, what am I going to find? My reaction is going to be a blue-black color change. So now what do I take? Again, I can take any food. All right, your teacher will probably put out some potato or some bread. Again, we like to try milk, or we could even try sugar. All right, because if it doesn't change color, then we know that there's no starch in it, if it remains brown. But if it does change to a blue-black, then we know that starch is present. So there we have done our carbohydrates. Okay, now we're going to go on to our proteins. I'm going to move it over here. And for my proteins, I've used, what do we know that's in proteins? I've used good old egg white, all right, the white of an egg. Now I'm going to use Millen's reagent, which is very, very poisonous. And generally, most schools don't use it. All right, so if you do, you know the general rule, all right, never ever touch something or smell something or take a whiff of something that you don't know. Some of this Millen's reagent, or you will find some of you, all right, there's two tests that you can use. The other one is called the Biuret test. And the Biuret test, you use combination, all right, of sodium hydroxide and the lovely blue, see, blue is such a lovely color. All right, the lovely blue copper sulfate. Right, so when it comes to the protein test, you have two choices. One's the Millen's reagent, all right, and the other one is the Biuret test, where you use sodium hydroxide and copper sulfate. Okay, once again, obviously beforehand, I put it in my water bath. Wow. All right, now have a look at this change over here. Now, I don't know if you can see there, okay? If you have a look, when it comes to proteins, the color here, we have a violet, it turns like a purple color. If you have a look all around here, all right, you will see even at the bottom, it is purple, all right? We say it's violet or it has a purple precipitate. So you see the color change here, all right? There wasn't anything. There's my Millen's, and it was see-through. Even if I put my copper sulfate, it would have been blue. And look here, a lovely purple forms around it. Also, what you should have noticed, if you have a look at my egg, okay, James says you want to see the lovely purple again. Can you see that? Can you zoom there? All right, there we go. We see all around there the lovely purple color, okay, a purple precipitate. Now, going back from last week, I put this egg, which is a protein, into boiling hot water. Now, if you can remember from your proteins are sensitive to temperature. And when they put into very hot water, they denature, they change their shape, right? And remember, enzymes were proteins. So if you have a look, here's my egg, all nice and, all right, I don't like eggs, and egg is never nice. Okay, here's my egg, all floaty, and this just looks gross, all right? All lumpy and cooked because it's changed its shape. Okay, which leads us now to the last one. I won't say it's the most boring one, but no big color change happens here, and that's to test for lipids, all right? And I'm going to take good old oil, all right? They are good for oil. Going back to the proteins, again, as I said, if I test this, now I can take my milk powder, I can take peanuts, I can take whatever food that I need, all right? And if you're going to use big chunky pieces, always grind it up or cut it up or use the pestle and mortar and grind it fine, fine, fine. So it's much easier, all right, for the reaction to happen. Here we're going to take a little bit of oil. All right, I think, here we go. Let me put it in quickly. Oh, okay, I'm going to use my teeth. I know my mother's probably saying, you never use your teeth. All right, so I'm going to put a little bit of oil. Now, I want to actually make us like an emulsion. I can't say solution because we know that oil doesn't dissolve in water. All right, so now what I'm going to add here, all right, nice big, I'm sure you've all got it, ethanol alcohol. Okay, I'm going to put the alcohol right with the water, with the oil, and there you'll see already, all right, it starts to make a solution. Now you need to just shake it up a bit, all right, because if we go back to last week, all right, one of the properties? One of the properties of oil was that, all right, one of the properties of oil is that it doesn't, all right, mix with lipids, it doesn't mix with water, but it does dissolve in alcohol. So now you'll see over here what we've got, all right, is a solution, all right, of oil and of ether, alcohol. Then you take filter paper, right? You take filter paper, you take a little bit of the, all right, solution, and you pour it onto the filter paper, all right? And then you wave it in the air for a while, and you wait, and then voila, what you're going to get. This is the least dramatic of all the responses, okay? This is what we call a translucent, greasy spot. Some of you have got lots on your clothes. I know I do. I always land up spilling my food somewhere, all right? So we have got a translucent, greasy spot. That is the result, all right, at the end of my lipids. So for lipids, what did I use? My reagent was ethanol alcohol. You can use formalin, all right, any kind of thing that the alcohol will dissolve in, or the oil will dissolve in, all right? What is your end result? A translucent, greasy spot. So just to recap, the blue, for test for glucose, the blue will change to a brick red or an orange if glucose is present, right? The starch will change to a blue-black using iodine, all right, if it is present. The proteins will have a violet or a purple precipitate or a violet tinge, a purple tinge, right? And we use millins, or in the biuret test, we use sodium hydroxide, all right, and copper sulfate. And then lastly, for our lipids, we used ethanol alcohol or ether, all right, and what our result was a translucent, greasy spot. Okay, Luni, are we ready for a break?
Yes, we
I
can.
think so, 'cause we need to get that challenge question that we forgot
All right.
out. Yeah.
Mindset is, let's take a quick break. After the break, she'll help you guys with the challenge question. Just go through it, how to do it, and all of that stuff. And we'll see you then. Welcome back, Mindsetters, from that break. I hope you guys didn't miss the beginning part because it was too cool and Cheryl was doing food tests and all of that. Anyway, quick shout outs to Mandibu, Mash, Lungelo, Moya Habo, Tepiso Sihle Banyana, Samge, and Meshach. Thank you so much, guys, for tuning in. Now we are going to tell you about the challenge question, which will be posted on our Facebook page. So if you just go through it, answer it in the comment section, and then you will be fine, Cheryl.
I'm sorry, I really should have done it right at the beginning, but I was so excited to do the food test that I just totally forgot. Also, when you get to my age, right, the brain doesn't always work as well. Okay, the challenge question, it's a multiple choice question, but you need to listen very carefully. I'm going to go through it with you, all right? A student was asked to identify the two compounds in each of the 3 test tubes. The table shows the result of the test the student performed on each of the compounds, all right? So you've got 3 things, test tubes X, Y, and Z. Here is the test, all right? The sodium hydroxide and the copper sulfate. Let me get to my pen, all right? So that would be test for protein, Okay, remember, carbohydrates, right, and iodine for starch. Okay, and those are your results. Okay, so that would be the different color change. Now it's multiple choice. Which conclusion is consistent with the results? Right? Egg white and sucrose were in tube X. B. Peanuts and starch had been placed in test tube Z. Maltose and sucrose had been placed in test tube X. And starch and sucrose had been placed in test tube Y. All right, we'll come back to those questions just now at the end. See it, just a multiple choice, but see if you can work out how you got to that answer. All right, just a quick little summary. All right, I've got 3 words on the board, all right, and all that I need you to do, it's very important for this before the food test. When I was dealing with it just now, you saw what I put emphasis on, all right. So for each of them, I want to know, all right, number 1, okay, what am I testing for? So when it comes to carbohydrates, I'm testing for glucose, all right. What do I use? I'm going to say Benedict's solution, okay, or Fellings. Both of them were blue. Fellings A and B. You'll see there are 2 things that you add together. What is a positive result at the end? It would be a brick red or an orange. All right, color change. So there we go. What am I testing for? What reagent am I using? And what would be a positive result? Okay, so if I test for glucose, I can take any food. And if I follow that whole process, very simple. At the end, if it changes to one of those, yes, glucose is present. If it stays a blue color, then we know no glucose is present. Except, all right, some of you might do the sucrose test. Okay, to test for sucrose, sucrose is a disaccharide, all right? Now, sucrose is the nice sugar that you have in your cereal or your table sugar that you have in your tea or your coffee, okay? That is sucrose. Now, sucrose, all right, when you eat it, believe it or not, in your stomach, it gets broken by hydrochloric acid to produce glucose. So if you were to do the glucose test, so if I took sucrose and I added Benedict's solution and I put it in a water bath, nothing would happen. But if I take sucrose, right, I add hydrochloric acid to it. The hydrochloric acid breaks it up into glucose, all right? And that glucose, if we add the Benedict solution and we heat it up, it will change to that color. So when it comes to the sucrose, we do need to use hydrochloric acid first because it breaks that sucrose up into the 2 glucose molecules. Okay. Then for lipids, what are we testing for? We were testing for fat. Okay, let's say, hang on a second, I'm jumping the gun here. Carbohydrates. All right, starch. What do I use? Iodine solution. All right, be careful, not just iodine. Iodine solution, and a positive test is blue, black. All right, that is a positive test. It's brown. If it stays brown, you're not going to use it. And this is important, all right, because you're going to do photosynthesis. And when you do photosynthesis, you need to see if starch is the end product. So you're going to test with iodine. The same for glucose. For example, in grade 11, when you do the excretory system, the urine, you can do a practical, all right, where you can perform a glucose test, and if the person has glucose in the urine, you can do a simulation. You don't get people to urinate, don't worry. Right? You can do a simulation that if there's glucose in the urine, which you've performed a glucose test, you can then come up to the conclusion that maybe this person has got diabetes. So knowing your food test is a basis for a lot of the things that you're going to do, especially when we get to enzymes, etc. Because remember, enzymes were proteins. All right. Then I had my lipids, my fats. What did I use? All right, I'm going to say here I used ethanol alcohol. All right, or some of you have got ether at school. All right, and what, I'm going to this side because it's quite a long word. A translucent greasy spot. Okay. So when it comes to lipids, it's a translucent greasy spot. When you put the water on the filter paper, you'll see water will make will change the filter colors paper slightly, but it's not going to be to the extent that when we put the oil on and you keep waving and you put that filter paper up to the window, right, you're going to see like maybe a wet mark around where the water was, where it's like a water wet mark, but you're going to see this greasy spot, like, after you've had the chips and all your grease on your hands, etc. That's exactly what it's going to be. Then for our proteins, all right, at the bottom, okay, there's two tests. For proteins, you can use Millen's, okay, or Millen's reagent, all right, or you can do what we call the biuret test. And you'll see the challenge question, that's where we did it. All right, what do we use? Sodium hydroxide and copper sulfate, the nice blue one. And often you can use these copper. Oh, okay, now I'm just going to, sorry. I know I've got an eraser, but copper sulfate. Right, and what are my results? My results for proteins, all right, it changes to a violet color or a purple, all right, or if you saw there with mine, okay, we could say a purple precipitate. What do we mean by precipitate? It means it was around the edge. Okay, so that is very important. For each of those, you're going to see one of the questions now that we ask, right? When it comes to the food test, the questions, it's not a very intense questions to it, all right? You'll see the questions that I've chosen. You need to be able to know what you did, but most importantly, what was the color change? All right, what was the color change? So now let's start with our questions. Question number 1, all right? The pie charts below show the composition of 3 different organs from the body of an ox. The figures are percentages. So here, all right, if you have a look, we've got what they were made of. So we've got proteins, minerals, lipids, and carbohydrates. Now the question is, describe how you could demonstrate, all right? Describe how you could demonstrate that a sample of heart contains protein, right? What are they asking you here? They're asking you, how would you test for proteins, right? So you actually have to put a method down, all right? And when you do a method in the practical, you need to write it in order, okay? And you need to use bullets. You can't just write a paragraph and put this before that and whatnot. It has to make sense, all right, for 6 marks. So what are we going to do? All right, let's have a look. Let's extend the page. All right, what's the first thing that we're going to do? The first thing that you do is you cut, right, small pieces of the ox heart, all right? Isn't that what we need? Okay, you could say cut, or you could put over here, you could use the word grind. Remember what I said to you, and those of you, if you go and have a look at the notes as well, it says to you, all right, that you must, if you're going to have like those solid pieces, you must cut it up finely, chop it up, or grind it up so that, all right, that it can be saturated by the chemical that you can use, so the reaction can happen quite quickly. All right, then what do? We place number 2. You don't have to do number 2, or you can just put bullets. Place the ox heart pieces in a test tube. Okay, now it will depend on, all right, what do you have? Are you going to use the Millen's, or are you going to use the biuret test? What I would suggest before you do that is to put a little bit of water, all right? Place some water, not a lot, in the test tube. Okay, in the test tube. Now, what is the next step? Step number 4. Let me see, can I go even more? Yes. Step number 4. Now I need to add my chemicals. Okay, so you're going to have a choice. Add, and now you can choose. Are you going to use Millens, all right, or are you going to use sodium hydroxide and copper sulfate, all right, to the test tube? And then what you do is you mix it, all right? Mix with a stirring rod or a glass rod, all right? You don't want to, stirring rod, you don't want to contaminate it. Then what do? Number 6, place test tube in a water bath, all right? So you put it in boiling water, and then you wait for the result. What will your result be? Then number 6, okay, when you take it out, if there is a purple, all right, precipitate, or violet, or depends on what you used, right? So if it changes color, if that little mixture that you made, almost like that you ground all the meat together with the water with the chemicals and you put it into, all right, your water bath. If the color change is purple, violet, you can then conclude, all right, then number 8, this is your conclusion, all right, the ox heart contains proteins. Isn't that what you tested for? All right. If it didn't change color, then your conclusion would be that the ox heart does not contain proteins. Go back to the top of the question. All right. It asks you, describe how you could demonstrate that a sample of heart contains proteins. So remember what I said to you, that protein test that I did there with the egg, that is your experiment. All right. That will always give you a positive. Now we want to find out. So if you were to do that practical, instead of putting the egg in, crush up the ox heart, and we add exactly the same reagents to it. We add the copper sulfate or the sodium hydroxide, or we add the Millens, all right? And we wait for results. And what the result is, if it's violet or purple, yes, proteins are present. If that is not, if it doesn't happen, then we conclude no, proteins weren't present. They didn't ask you yes, no, they wanted you to know, demonstrate how would you test for it. And basically we're asking you, how do you test for proteins? So instead of you can't add the egg white, because that's not what you were testing for, right? They wanted you specifically to then substitute that egg white with the ox heart. Okay, next question. This is, the writing is a lot more than, all right, it's a bit of a misleading question, because actually what you need to do is actually quite simple in the end. You can use you can take a lot more questions from this graph, and you can put it into the biochemistry. You can ask about why do babies need to have proteins in their milk, etc. But I've just taken the food test component out of it. Okay, it says here, milk provides a young mammal with all the nutrients it needs for its early growth and development. Milk contains proteins, carbohydrates, and lipids. It also provides essential vitamins and minerals. Can you see how we can branch this question out, right, to the biochemistry, vitamins, and the minerals that we did last week? It also, okay, the composition of milk, however, varies enormously. It varies from one species to another, and from one individual to another, and even within a single individual. The graph below shows how the concentration of triglyceride, if we were to expand on this question, all right, triglyceride, fat, lipid, all right, lipid. Okay, so as I said to you, I'm going to concentrate on the food test part of it, all right? And you can actually, for the teachers, you can expand on this. Why do we need fat? Why do we need the proteins? Why do we need the carbohydrates? All right? In human milk changes, at lactation, the period of milk production progresses. All right? And what basically this graph is showing you that the fat content of breast milk, basically, there's quite a high fat content, but we also know proteins, all right, minerals, and vitamins. Now, as I said, to test your skill, what they're trying to say there is that it is everything that a baby obviously would need to be able to grow and develop. And if we have a look again at last week's one, when we looked at carbohydrates, lipids, fats, vitamins, and minerals, all right, we know that those were all needed, right, for an individual to be able to grow and be healthy, right? Because if they lacked any of those, we saw that we had your deficiency diseases, etc., your rickets, your kwashiorkor, your night blindness, your pellagra, all right, your scurvy. Okay, so just keep that in mind. But I'm going to, just because we're doing food tests today, right, it says here, the preamble states, and that means just the beginning, the introduction. Milk contains proteins, carbohydrates, and lipids. Draw up a table in which you state the tests used, the expected results, and a conclusion of your results. Okay, now let's have a look over here. Let me see if I can go again. Okay, for my table, no. Right, so it says you need to draw up a table in which you state the test used. Right, so start off. The first thing you need to do is you need to draw a table. And a table always has column, and they've actually given you the column headings. So, test for, all right, the expected results. Okay, so you could say expected results and conclusion of your results. All right, so when they say expected results, I'm going to put here what color changes. All right, there we go. Yeah, it worked. So all that we're doing now is we're putting exactly what we've been saying in a table. It says that the milk contains proteins, carbohydrates, and lipids. So what's our first one? Glucose. Okay, isn't that a carbohydrate? Okay, glucose is a carbohydrate. It's a monosaccharide. What do we expect to happen? All right, there we go, the brick red or the orange. What can we conclude? All right, that breast milk contains glucose. It's not too difficult stating the obvious, but again, putting things into a table, right? You put your independent variable here, the one that you are controlling. Right, you can also test for starch. Expected result would be blue-black. All right, and what that can you conclude? All right, here that breast milk has starch, but yeah, you need to maybe use your not but unlikely to occur. All right, babies at that stage can't really, all right, digest it. There we go. What's the next one? Here we go, proteins. All right, what's the expected result? Violet color change or the purple precipitate? What again? Sorry, I'm writing so it's the same thing over again. All right, what can you expect? Breast milk contains, all right, proteins. And then your last one, and that's the one that your graph, right, definitely, all right, says, and that is lipids. What will change? A greasy spot if you were to test it. And what can you say? That breast milk contains, all right, fats. So from that, what can you conclude? Right, it contains everything that a growing individual will need. Are we taking a break again?
All right.
I see the big zero there in front of me, and my earpiece has fallen out. I have no idea where it is.
Okay. My set is while she also sorts out her problems of earpieces and all of that stuff. We are going to take a break, so don't go anywhere. We'll see you straight after this. Welcome back, Mindsetters. From that break, we are going to do the challenge question now, so I won't waste any more time. Cheryl.
Okay, let's now go. I think we're doing it in the right order now. And I'm minus an earpiece because it's either my earpiece or it's me and the smartboard. So I think I'm like the problem child here. All right, let's have a look at our challenge question. Right, it was a multiple choice question, but basically the premise of it was as follows. Okay, if we have a look here, if you're not too sure, if we have a look, I said this was a protein test. All right, so now if it went violet, that meant X, yes, there was a protein in it. If it went brick red, yes, one of the things must have been, all right, glucose, all right, or you could have even said sucrose, all right, and iodine went brown, so it does not have starch. Okay, so that's in number Y. In X. Number Y, it went blue, so no protein. Benedict's solution, it went blue, so there's no sugar, and it went blue-black, so it means yes, there's a starch in it. Right? Number Z, right over here, violet, yes, we have a protein. No, we do not have a sugar, and yes, we have a starch. All right, so let's have a look. It says here, egg white and sucrose are in test number X. Look here, egg white, is it a protein? Yes. Sucrose, let's just keep that as a, all right. So yes, we have sucrose has been placed. Just keep it as a question mark, right? Because yes, that could be it. Peanuts and starch in Z. So peanuts, all right, and starch, all right, have a look. Peanuts, did we test for oil? No, all right. Starch, yes, there we go. So no, all right, we didn't test for that. Maltose and sucrose in tube X. Maltose and sucrose, no, because what did we have? We had something with a protein, right? And maltose and sucrose, maltose, no, right? We need to digest it a bit more until we can get it, because it's actually, all right, was once a starch, maltose broken down, but we can't test that. So we know it is a no. And starch and sucrose have been placed in test tube. Why? A starch, yes, and a sucrose, no, because there it's blue. All right, so egg white and sucrose in number A, that would have been your answer. Okay. All right, I think you've got some questions.
Yes.
Yeah.
So this one is from Lungelo Patient, and he or she is asking because they don't have a profile picture. A disaccharide is made up of 2 monosaccharides. Does this mean that 2 glucose form a sucrose and many glucose form a starch in a polysaccharide?
Yes. Perfect.
Thanks.
All right. Yes, that's as simple as that.
Okay.
Yes, a monosaccharide, simple. That's your glucose, your fructose, your galactose. Your disaccharide is when you have 2 monosaccharides together. Remember, the word tells you what it is. Di means 2. And then when we put a whole lot of glucose, all right, glucose is the monosaccharide that we use in polysaccharides, and that is your starch and your glycogen. All right.
Thank you. And then Lindo Musa Mabika is asking, when testing for protein, must you add both sodium hydroxide and copper sulfate at the same time, or must you choose one?
No, you don't. You do add them both. You don't have to do it at the same time. You can take one, all right, the sodium hydroxide, you put about a teaspoon in the copper sulfate, you just put a few drops in, right? You don't have to put it at the same time, but yes, you mix it together. You can't choose one, right? You can choose whether you want to do the Millen's test or the biuret test, but the biuret test, those two come together. They're not separated. Sodium hydroxide, copper sulfate, together.
Okay. And then Banyana Mahopan is asking, what's the difference between iodine and iodine solution?
Okay, iodine solution, all right, is, you know what, we actually, I won't say there's no difference. Then when we talk about iodine, we're actually talking about the solid, the iodine in salt, the sea salt, all right? Remember when we were looking at micronutrients, it's the chemical, the solid that we find in sea salt and seafood. Iodine solution is when it's in liquid form. All right, so it's been processed. That's why we use the word iodine solution, because when we use the word iodine, actually refers to the micronutrient that is necessary.
All right, and then
Okay.
2 questions from Tatenda Manenji. First one, what is the chemical name for iodine solution?
It's iodine solution.
And then the other one, what is the formula for carbohydrates?
Okay, now the formula for carbohydrates, be very careful there, right? When we talk about the formula, we really specifically look for the formula for your monosaccharides, all right? And if you can remember, all of them were CH and O, all right? And for the monosaccharides, it was N, 2N, and N, all right? So for example, if it was a glucose, it's a 6, a 12, and a 6. When you get to matric, you're going to learn about RNA and DNA, and they're a pentose sugar. So it's a 5, a 10, and a 5. You obviously get triose sugars, which would then be a 3, a 6, and a 3. Okay, that is for the simple monosaccharides, yes, for the carbohydrates.
And then QT Samdelays is asking, so can other tissues in the body also be tested for proteins or lipids?
Yes.
Okay.
All right.
Another one from Lindu Musa Mabika. When I test for lipids, can I use any type of alcohol?
Yes. No, you don't. No, you can't use any type of alcohol. All right, because remember, a lot of them are sugar. You need to use lab alcohol. That's what we call it, ethanol alcohol. All right, or ether, because the rest. Remember, the alcohol that's almost like pure alcohol. The other alcohol, if you're using wine or whatever, or brandy or spirits, has a lot of sugar in it. Okay, and because that's how they make it. So that will the experiments won't come out that well.
Okay, and then Mulisa Bugana is asking, why do we test food?
Why do we test food?
Yes.
Okay. Remember what I say to you. It's going to show you guys need to have a practical knowledge, all right, of things that are happening. So let's, as I used earlier, all right. If I ask you, if I put, they're not urine samples, but I do. I put urine samples, right? I say they're urine samples, and I ask you to test. I want you to test for glucose, for lipids, and for proteins. Now, you guys are going to do excretion, right, in grade 11. And one of the things you're going to know is when urine, all right, in the urine, one thing you shouldn't have is glucose or proteins. If you do, that means that there's something wrong. So if I give you a practical, I say to you, here, this is this person's urine sample, test it, all right? And you see, oh, this one's got glucose in. Oh, that one's got proteins in. It's not the food, we call it food tests, all right? It's not necessary that we need to know what's in food. We need to be able to, can we test for a lipid? Can we test for a protein? Can we test for a glucose? As I said to you, you guys are going to do photosynthesis, all right, when you do the practicals. And what do you need to know? You're going to do a test where you need iodine. And what does iodine do? Iodine changes color if starch is present, all right? And again, where would you have known that? You know that from the basis of your food practicals. So we call it a food test because that's basically what we're testing is food. But remember, those things came to be in food through processes. So we have glucose because the plants were able to make the glucose. We can test for proteins because, all right, plants can make the proteins and we can eat the proteins.
Okay. And then just a quick question from Sam Gendlela. She is asking, she doesn't understand how you got A and not C in the challenge question.
You know what, I was actually telling her, as I was going through the questions, all right, if I thought about it, let me go back. I actually think I would have accepted, oh, I'm going too far. I think I could have changed C as well, because if I looked at it, all right, okay, no, I would have got, you could have also had B. Definitely not C. All right. Maltose and sucrose, all right, if we have a look there, if we have a look at maltose and sucrose, both of them, all right, are disaccharides. All right, and if we go back over here, all right, and we have a look at the X, all right, the thing was, for maltose, all right, The maltose is a byproduct from starch. So if you have a look over here, starch was a negative. So that's why we know that it wasn't maltose. Okay.
Quickly,
All right. Yes.
what do the prefixes mono, di, and poly mean?
Okay. Mono 1, di 2, poly many.
All right. Is there time for one more? No,
time for one?
no. Chaile. So says James.
Is James talking in your ear? Yeah.
Thank you so much, Mindsetters, for tuning in. Thank you for a great lesson to Cheryl. Thank you for giving us an awesome lesson. Thank you once again, guys, and we'll see you next week. Make sure you tune in next week as well for an awesome lesson. From us, it is goodbye.